It's my enormous privilege this morning to introduce my old friend Huanming Yang, the president of the Beijing Genome Institute. I met him in Brussels at a meeting organized by Dr. Cunningham. It was entitled Genetics and the Future of Europe, and it was promoted by Monsieur Busquin, the commissioner. Can you imagine that one of the leading political figures in Europe felt that genetics was important for the future of Europe? And I met Huanming, and he spoke in the most brilliant fashion. And I can assure you that this morning you are going to see something exceptional. It was like a bolt from the blue for me. This is the man who started genome science in China. He had an opportunity somehow to persuade people that China should take part in the Human Genome Project. And in approximately a year, he built this extraordinary institute in Beijing, the Beijing Genome Institute as it was called. And they contributed a significant percentage of the human genome data. Not only that, he led the fight to keep the human genome data in the public arena. I think you all know there was a large row. On one side, essentially, there was American capitalism, and on the other side, there was public science, international public science. And Huanming Yang spoke out firmly and pointed out how important human genome data would be for everybody in the world. It must not become just owned by one or a small number of corporations anywhere in the world. His institute has grown extraordinarily. BGI now has 4,000 staff in laboratories in Beijing, in Shenzhen, in Hong Kong, in Shanghai. There is BGI Europe in Copenhagen. There's BGI the United States in Philadelphia and at UC Davis. They have produced about 1/3 of all the genome data produced so far on the planet. Truly an astonishing achievement. And I went to the 10th birthday party of BGI in Beijing, and what a celebration it was. A great personal celebration for all of those young Chinese people. This huge wave of young Chinese scientists who were brought into this by Huanming, nurtured, educated, given skills, and now they're giving it all back to us worldwide. They sequenced rice for obvious reasons, but they sequenced the silkworm. Just imagine that. Nobody, I think, in the Western world was thinking that that was important. They sequenced the silkworm. They took a major role in the analysis of the SARS epidemic. They've made countless contributions to science. So Huanming, one of my dearest colleagues, a person who's had an enormous personal effect on me, and I think you'll understand why when he gives his talk. Huanming, it's an enormous pleasure to invite you to speak here, Dublin, City of Science.
Thank you, David. Of course, I thank you for your brief introduction, which has even made me embarrassed. You have to remember, I am a student of yours, also student of Paddy, just like many other Chinese scientists who have been trained in Europe and in the USA. And then to respect the teachers is one of the cores in Chinese culture. I will never forget you. That's the reason. Please regard this speech, even not that excellent, even not qualified as a gift, but I really would give you as a gift for your continuous encouragement, fatherly advice, and the instructions in the last decades. Of course, as well as your great contribution to the global sciences and the international collaboration. That might be just a tale which I was told at my childhood that this century, at that time we call it the next century, 21st century, would be the century of biology. Many people don't believe it. But I was so convinced. I have to say, it did help shape my career of my life. Then I have been working in this field for a long time. Perhaps this term is not strong or attractive enough, or you can see there are few audience here. Of course, I especially appreciate your joining me. My talk will begin with the DNA double helix as well as the Human Genome Project. We are actively planning the global celebration of the 60th anniversary of discovery of DNA double helix, as well as the 10th anniversary of the official completion of the Human Genome Project. What would be the major impact the HGP played? I would say that's omicization. I would like to quote from one of our colleagues: within the next 25 years, omics will be the biggest, if not the only game in town, also in Dublin. It's not an easy job. Just listen to one of our colleagues. We should never know the complete base sequence of the human genome. If we knew it, I can promise you it will not be known for the next 3 years. No, not long enough. 30 years, still not enough. We have to wait. We are asked to wait for 300 years. It's not true. Science cannot wait, and this issue is also so important. We have had it done. More importantly, we have had it done together. That's the first international project which was carried out or completed by both developed and developing countries. There are always two sides to everything. You can see more than 3,000 researchers or technicians in 16 centers in 6 countries, and it took them 13 years at a cost of $3 billion just to finish. Actually, it will never be really finished, to nearly finish the genome of a single individual of mankind. The situation is quite different now. The same job could be done 100 times on a single day in a single center like BGI. Let's celebrate this revolution. Let's celebrate the advancement of science. I'm very confident which will bring all we need. Until now, actually, as of the end of last year, BGI, my institute, had generated about 650,000 gigabases of sequence data. Please be reminded that our genome size, it sounded so huge just a dozen years ago, was only in the size of 3 giga. It's about 200,000 human genome size. If we do something, especially for us conservative Chinese, you should have a belief. You think you are correct. You think you are right. True, there are 2 beliefs in genomics. The first is life is in sequence. Please allow me to quote from Dr. James D. Watson: the precise sequence of bases is the code which carries the genetic information. Then the second belief is life is digital. Perhaps it's not so easy to understand. Again, allow me to quote from Sir John Sarsden in UK: the instructions for making a life from one generation to the next is digital. If we put these 2 languages together, the natural language for life, and then this, I don't know whether it's artificial language for information sense, we can see life in nature is digital. Based on these two beliefs, and then we have two technical platforms, sequencing machines and computing machines. But don't believe that you can do your jobs well just with the hardwares. For example, we have the huge computers, and we also are in collaboration with those huge computing centers in China. More importantly is the softwares. We have developed numerous softwares. Of course, not every of them is good, but still some of them are evaluated by the international community as best of the best. We have received several awards related to computing, especially cloud computation. We just gave a demo for the data transfer. Generally, if we transferred it by the system of public internet, it would take 1 day, but with our system, developed together with our colleagues, it would take just 30 seconds. The world is becoming even smaller. I just hope the smaller world would give us more confidence of the future. And then Human Genome Project also cultivated a new culture for scientific research. That's the culture of collaboration. We all realize in our community that genetics is international. Genetics cannot be done alone. Please again be emphasized. All the jobs I will talk about today would not be done only by my institute, BGI. It cannot be done without the vast collaboration of international partners. And more important, all the culture was on the spirit of the Human Genome Project. That's to share. In other words, that's really free lunch. We're making free lunch. That so-called the future will be owned by all. All the jobs should be done by all, and all the achievements should be done by all. I'm so lucky that I have so many friends all over the world, just like what David told me. If we don't have a friend yet in a country, we can immediately make it through our colleagues. I'm so privileged that I have so many teachers or mentors from whom I have learned so much, and then at least 5 of them are among you. Everything has its history. The sequence technology might have made 3 breakthroughs in 3 stages. The first, we all know, that's from hand sequencing to automatic sequencing. Again, as any other breakthroughs in science, we took the opportunity. And China became the latest contributor to the Human Genome Project and placed itself alongside France, Germany, Japan, UK, and USA. Even our contribution was so small, it could not be smaller. That was a breakthrough. That compared to what we have today, it was really a poor technology. For example, our task given by the International Human Genome Sequencing Consortium were only 500,000 reads. And then for every sequencing read, it would take 30 tips in half a year. We used up 15 million tips. It's almost unimaginable if we just counted how long it would take. And the second breakthrough, which is important, which contributed to the completion of the Human Genome Project, that's from slab electrophoresis machine to capillary electrophoresis machine. We have also taken the responsibility by completing the draft of the rice genome. We all know rice is so important for many people in the world, especially for Asian people and for Chinese. If we say, what about the good job? First, the paper should be published in a good journal. Yes, we publish it in Science as the cover story. And Science magazine came to Beijing to have the press conference to congratulate or to honor Chinese scientists to have sequenced the rice genome. And then the second, it should have a lot of citations. It is true, until now, this paper has had at least 1,500 citations and still being cited now. And the third, evaluation by the scientific community. It is true, it has given the highest marks by faculty of 1,000 and regarded as a landmark paper which should be read by all plant biologists. And what about its real impact on research? Let's look at this figure. Before the rice genome was published, there were more research on wheat than on rice. It's even more significant for those developing countries. And then, how to interpret it? Our colleagues in the International Rice Research Institute give me this slide. The reason is only with the genome sequence, and then the researchers, the breeders are able to clone those genes or QTL related to high yield and high quality. And now, of course, we're also working on wheat. From here, from this example, we can see that the genome sequence is just the beginning of research or use of any organism, and genomics is the basis of life sciences and biotechs. We're laying the foundation for a tall building. And of course, still softwares. And then we made these softwares called RIPS. It has been published. It has been released many, many times, used by many labs to solve one of the technical bottleneck of whole genome shotgun sequencing technology to make rice the first really completed by whole genome shotgun strategy. The situation is again changed. At that time, actually, we only sequenced one of the strains of rice. Now we're going to sequence 10,000 rice cultivar or accessions or strains. The third breakthrough in sequencing technology is even more important and has had more important impacts on the whole field of life sciences. This is from the traditional sequencing technology to massively parallel sequencing. That means we can sequence a lot of DNA molecules at the same time. Now the number has reached 1 billion. We took this opportunity. We completed the first Asian individual genome, and it was also a cover story in Nature. Scientifically, that paper told us with sufficient data that human genome diversity or the difference between different populations would be much bigger than we expected. But more importantly, technically, as reviewed by science, this paper indicated that the methods now, or the sequencing technology now, are extremely powerful. That's the time we called for faster human genome sequencing as a call to all our colleagues. The real breakthrough made by us, of course, BGI, together with its colleagues, is the first genome sequence of the giant panda without any help of so-called genetic map, without the help of physical map, without any knowledge about that technical bottleneck, unknown repeats in the genome, were able to sequence and then de novo assembly of a large genome without any further information, just by sequencing technology. Of course, we owe this success to the softwares written by our young staffs. After taking the opportunities, BGI is poised to become the biggest DNA sequencing laboratory in the world, as said by economists. And then BGI Shenzhen enhances its reputation as the world's large sequencing center by doing this and that. in the last 4 years, we have published in collaboration with our colleagues about 6 papers in Science, in Nature, its series, in Cell, in New England Journal of Medicine, and other important or influential journals. That's our contribution. All the data were generated through our platforms of sequencing. Now, until now, there are about 2P. 1P is 1,000 T. 1 T is 1,000 G. And then we have generated so much, so our contribution, that's as of the end of last year, at least 1/3. It's still not enough. I would say sequencing technology is a powerful tool for all biologists. It's an opportunity for us all. I'm very confident that every corner of life sciences, every lab definitely would use the genome sequence data because it's just the beginning, just the first step, just the basis or foundation of any research in life sciences. What will we go? There are so many directions. I just give you 4 examples concerning the trends, concerning the direction. First, to sequence more species. We have published all in the important journals, and then 2 monkeys, many CHO cell lines, a naked long life mole rat, and the 2 species of ants. And then we just published the genome sequence of yak. At least we have answered the questions related to adaptation of yak to life at high altitude. Based on these achievements, we have initiated a big project to sequence at least 1/4 of all of the known vertebrates on Earth. And then for invertebrates, we have just published these 2 worms, both in Nature and Nature Genetics. What about plants, which are even more important to us, or equally important? We published that cucumber, and then potato, a Chinese cabbage, Indian pigeon, pea genomes, which all have been used with the data for breeding. And just 2 months ago, we published the tomato genome in the name of the International Tomato Genome Consortium. It's also important for us, even though it is named foreign eggplant by Chinese, because we know how to acknowledge the intellectual property right of yours, because China now is already the largest exporter of tomatoes as well as second largest producers. This is an important paper. In collaboration with our European colleagues, that we found out there are at least 2,000 species of bacteria in our gut. They are working day and night. Then every of us would have at least 170 species of bacteria in our guts. What about the genes? In all, at least there are about 3.3 million genes or open reading frames. This number is about 150 times of the estimated number of our own genes. Now, we should not exclude these genes. Our genome should include other genomes. That's the bacterial genomes, which I will mention later. And you all know the pathogenic E. coli in Germany last May. We published finally with our colleagues in New England Journal of Medicine. We also published that HPV integration or its relationship to liver cancer. That's a vast collaboration. 3 major pharmaceutical companies, just like Pfizer, and then Merck, and then others have joined it to make this free lunch. Also based on these achievements, we initiated the 10,000 microbial genome project. Here, 10,000 means everything under the sunshine. This project aiming at sequencing and analyzing most, if not all of the characterized culturable bacteria or other microorganisms collected from the world. We also initiated the Earth Microbiome Project. This is different. As what we did for the bacteria in our gut to sequence the mixture of the sample, we are going to sequence at least 1 million samples collected based on the Google map from every corner of the world. Listen to our dream. Listen to our ambition. It's not only my ambition, my dream, that's the dream. This dream will be realized in the nearest future, that we are going to sequence everything or every living thing on Earth. We might say nothing in biology makes sense except in the light of evolution. Now we would say nothing in biology makes sense except in the light or with the basis of sequence, genome sequence. Then every single species you can think will be sequenced, not just one of them. That's the second trend, to sequence more individuals, much more of a single species. This job began with the International HappyMap project, which was the sister project of the Human Genome Sequencing Project. And then we have published so much, and China, mainly BGI, together with other two centers, contributed about 10%. UK and China, as well as our colleagues in the United States, initiated the 1,000 Genomes Project. Until now, we also have published so many papers in Nature and its series. This project is very important. It tells us that the complexity, the genome diversity of mankind is much more higher than we expected today. It was not a mistake in the past, just we did not have sufficient data to elucidate this important problem about ourselves. There would be two major points, I think very important to us, by the 1,000 Genomes Project. First, as I have already said, there are much more sequence variation. Now we call the single nucleotide polymorphism at least 15 based on the data generated by the pilot project, and now much more. If you know these figures, you can say now there are about 38 million of single nucleotide polymorphism and 1.4 million insertion and deletions, and then over 14,000 large deletions in our genomes all over the world in the human family. And then the second, many of the variations are population specific, and which lays the basis for us another big paper, human pangenome. We estimated that there are so much, about 0.6% to almost 2% of novel, of unidentified sequence, which are totally population specific, which might be present in the genomes of European, but absent from the genomes of either Africans or Asians. And then it also includes that individual specific genome sequence. For example, my genome sequence might contain some sequence which cannot be found from yours, and you also have your unique genome sequences. Even the function of these population or individual specific sequences should be further studied. It is true that some of the variations are related to evolution or adaptation. For example, published this paper in Science that we revealed that many Tibetan Chinese would have that allele of this gene, but it's rather few in the High Chinese, which could at least partially interpret why Tibetan Chinese would have adapted themselves much better to the life at the high altitude. That's also a big collaboration, that we sequenced more than 200 drug-targeted genes, which are important to drug development of 14,000 people. That's true. With or following more and more genome sequences are published, we'll know ourselves more and more. What about RNA editing? That's an old term. But with now new data, we publish this paper in Nature Biotechnology that we have found. It is true. Many RNA sequences would be different from its temporary DNA sequences, just because we have a huge amount of both DNA and RNA sequences. What about the figure? The figure is much bigger than we thought before, but still much smaller than another paper, which is so influential, published by our colleagues. How many people to sequence? Of course, a lot. Now we are going to sequence everybody in the world if you are fully informed, if you make the choice by yourself. At this stage, we are going to sequence in collaboration with our British colleagues, at least 10,000 English. And then for Denmark, we are going to sequence, of course, together with the Danish colleagues, that's how much? 50,000 of Danes. It's about 1% of the whole population in Denmark. And how many people to sequence in China? At least 1 million Chinese genomes. It would take us 30 years if we still have this capacity of the platforms. And then you would say, then is it such a long-term project? I can promise you, with the revolution or further improvement of the sequencing technology, this purpose, this target would be realized within a decade. I am confident. Then the third, from genomics to genetics, we are sequencing for more characterized phenotypes. This job began with the chicken genome. As many years ago, we sequenced that chicken for meal, chicken for egg, and chicken for medicine. And we found a lot of genome variations among different strains of chicken. Together with the big discovery, we sequenced data that most of the chicken now have its more than one origins. And then in 2004, we just sequenced one strain of silkworm, but now we have sequenced more than 100. We published 40 silkworm genome sequences in Science, and then all these strains have well-defined phenotypes for different color, and then for different yield, and for the quality of the silk. Corn or maize is important. We published the first 6 elite strains of maize, which have greatly contributed to maize breeding in history. And just a few months ago, we published another 2 papers in Nature Genetics, maize and maize. That's soybean. And Chinese cannot survive without soybeans. For both soybean juice and then soybean curd, that's tofu. We published at least 30 wild and cultivated soybean genomes. And then now we are going to sequence even more, at least 1,000 soybean genomes. And then we will find out why this strain of soybean is quite different from the others. That's the story about rice. We are going to sequence all the collected rice accessions or strains in the International Rice Research Institute. And we have at least published 50 of these genes. Or these genome sequences are related to high yield, high quality of rice. We still love rice. At last, and more important, it's the sequencing for application. Of course, mainly agriculture and the medicine. And for agriculture, we have made a lot of achievements on that foxtail millet. Actually, this millet is the unique crop which was domesticated by the Chinese ancestors and which has contributed a lot to solve the food problem in Chinese history. But now it's almost released by rice or wheat. We have made this data successfully for breeding. What about health? There are at least several directions. I would say that the genomics is the foundation of so-called personalized medicine. We have written a lot of reviews together with our colleagues to advocate the importance and acceptance of personalized medicine. And first, we published this paper and with all details about exome sequencing or sequencing of all the protein coding part. And then we reviewed that this method, which works so well and affordable, is a powerful tool for monogenic diseases. That's the reason we have published a lot of papers, was initiated 1,000 monogenic disease, or as the international collaboration. This is another report that the BGI and the hospital CHOP in Philadelphia plan to sequence at least 1,000 rare diseases, and the major part are monogenic disease. The second, cancers. Of course, I'm a layman on cancer research, but in collaboration with our colleagues, we have published the renal cell carcinoma, another paper on blood cancer in Nature Genetics. We all know that heterogeneity is one of the major technical bottlenecks in cancer research. Cancer genome research, we call the canceromics now. This is a typical slide from a cancer tissue. But if we use that modified single-cell genome sequencing technology, we can find actually this cancer tissue, this sample, contains all different cells, even normal cells and cancer cells at a different stage with different variations. We published 2 papers, one on the kidney tumor in cell, the other one on leukemia, also on cell. Of course, now it's still more expensive than the general whole genome sequencing. But this has spurred a lot of interest among the cancer genomics community. And the third is still about metagenomes, all the microorganisms in our guts. We published this paper, and then another one we call the enterotypes of the human gut microbiome. I'll just give you an example. That's the type 2 diabetes. We published this paper. That's quite another method, genome-wide association, or GWAS, in Nature. Then we analyze the microorganisms, mainly bacteria, in the guts of the patients or type 2 diabetes, as well as the well-selected normal individuals. Then we found there is a significant difference between type 2 enriched patients as well as the normal control enriched. The bacteria in the guts are different from those of the type 2 diabetes patients and from normal controls. Until now, we have identified at least 40 potential bacterial species which we think were relevant to type 2 diabetes. This paper has been accepted. We will see it soon. It is true. Our bacteria would, in collaboration with our own nuclear genomes, are working for us. Perhaps sometimes they would betray us. That's another issue. That's a disease. And then, based on the functional analysis, we also found it is true. The reason is these bacteria, they are functioning. They would interfere in the metabolic pathways, the signaling pathways, and gene regulation network of our own bodies. Last, not the least, is the early and non-invasive prenatal tests by partially degraded and a trace amount of DNA for sequencing. The discovery of fetal DNA fragments partially digested was made by Professor Lu in the Chinese University of Hong Kong
from mother's blood, and we immediately used the large-scale high-throughput sequencing to sequence that small amount, partially digested or degraded DNA molecules from the blood taken just from peripheral blood of the mother. It is fantastic. We jointly published a lot of papers. Until now, we have done more than 30,000 prenatal diagnoses by means of non-invasive genome sequencing. And then at least for the chromosomal numerical change diseases, it works very well, almost with 100 accuracy, without any detectable false positive or false negative results. It is under approval from the relevant authorities in China. It is immediately applied for the early and non-invasive prenatal testings, as we said, based on the principle of fully informed consent. I'm talking about trends. If we use the term, geographical term, direction, first, it's southern sequencing. That's DNA sequencing. This term is taken from southern blotting, as you all know. Then the second, northern sequencing. That's RNA sequencing. Now there are three major technologies: whole transcriptome sequencing, digital expression profiling, and small RNA or even long non-coding RNA sequencing. The third is still Western. That's the same like Western blotting. There are two major techniques: translatorome sequencing, ribosome protection, and regulatome sequencing. That's just the chromatin immunoprecipitation plus sequence. The sequence, just as southern blotting has its branches to all different fields of biology. We published this paper in Nature Methods, how to further improve these methods to use the sequencing for protein analysis. We know what southern is, northern is, western is, but what is eastern? Eastern is the first direction in Chinese culture. We mean methylome. We published this paper that's on the silkworm methylome as well as another paper on the human methylome sequence at single base pair resolution. We have just published this paper because epigenomics is tissue organ specific. That's a smart idea that they have analyzed the methylome of different various organs of pig and find out at least one gene which might be related to being fed can be or would be present in human genome. Now we are touching some basic issues in classical genetics, for example, identical twins. In collaboration with our colleagues in St. Thomas Hospital in London, we're going to sequence both the genome and the methylome of 5,000 twins. Let's wait. Let's see what will happen from the whole genome sequencing of the identical twins concerning both the genomes and the methylomes. If we look farther and wide, it reminds me again of that tale, century of biology. I was told at my childhood that the 21st century would be the century of biology or life sciences. Let's analyze it. Let's predict it at the molecular level, cellular level, tissue level, organ level, individual level, perhaps also ecological or biosphere level. Now we have all the methodologies at the molecular level. We all call the molecular methods to work with DNA, RNA, or proteins and on these cells, including GM. Then we have another technology, which is very promising, the stem cell and iPS. We know how to induce the stem cell into different tissue and the cells. And then now we can also reprogram those terminated, differentiated cells back into the cells similar to stem cell we call iPS. We have sequenced many cell lines of iPS, and we concluded the low incidence of DNA sequence variation in human iPS stem cells, and then we say cheers for iPS, definitely it will be widely applied in medicine. And then we, in collaboration with the GE, are going to sequence all the iPS cell lines GE has established, because to know the genome sequence or the genetic background of the iPS cells would be important to further application and research of the cell lines. We just attended the talk given by Dr. Craig Venter yesterday, and that's synthetic biology. Even though I don't agree with the term man-made life, but I would like to quote one of our colleagues. The program of life was written 4 billion years ago by somebody. Now it's time, obviously, for human being to rewrite the program. It was done. It was done by Dr. Craig Venter and his team. The first computer-designed, chemically synthesized, and together with biological methods of the genome was made. Even though it has the obvious mimic of the natural life, but we have to acknowledge this is the first man-designed genome. So I prefer to call it synthetic genome instead of synthetic biology. Just a few months ago, I made these predictions together with our colleagues that now we're able to make a genome of about 1 megabase. That's Cynthia. That's made by Dr. Craig Venter of Huanming. And then in 3 or 5 years, we're able to make a genome in the similar size of E. coli, about 5 mega. Then it would take us at least 8 or 10 years to make the first genome, which is eukaryotic, which is in the similar size to yeast, 15 megabase. Again, I'm wrong. Of course, now let's wait and see. We just announced on the 16th of April in Beijing that several groups from USA, from UK, from Canada, from China, and from India, and I also would like to have our colleagues from Russia to join. Before 2015, we are determined to design, to synthesize, to make a line of a genome similar to yeast. Still, I have to admit, it has all the traces of mimicking the nature, but we have done the modification or changes as much as we can. We delete. Now, in the blueprint, many of the repeated sequences, we have added a lot of things, perhaps just for fun, or perhaps it would have its significance in the descendants of that first man-designed synthetic genome of eukaryotic cells. That's sense. Sense will bring all the hopes, and then, of course, we have to realize clearly what the problems were created by synthetic biology, but sense itself in nature is ethical. From here you can see one of the major reasons for us to sequence so many organisms and their individuals. Now we are reading genomes. Now, finally, we will write genomes. So I regard synthetic biology as a natural development and a high stage of achievements of genomics. This is sense. Many people don't like it. That's really true. For example, we just announced that we have cloned another sheep, just like Dolly. But this one can produce unsaturated fatty acid. And one of my excellent friends, the students, and then Alan is also here. Of course, it's not strange and nothing new to clone a mammal or mammalian individuals after the birth of the sheep. Most, not all of the mammals have been cloned. But we have 2 modifications or improvement. First, we significantly simplified the methods for animal cloning. I can promise you, you're able to do it in your kitchen, not the kitchen, of course. And then the second, to combine GM. For example, this is something we are familiar, GFP. We can see expressed so strongly in the newly born and the adult pigs. And pigs has an advantage that we can get as many eggs as you like from the slaughter, and then also in evolution, especially now, I am becoming more and more similar to a pig. We have cloned numerous pigs, at least 500 pigs in our animal farm. This is the problem. This is the most famous pig in the world. It survived 36 days after that terrible earthquake. And now we have cloned 6 of them. I really don't know that this pig is called strong the pig will live forever through their clones, or actually they are the daughters of her, so never clone human beings. Of course, our major job is to make animal or pig model for diseases. This is good. Of course, we know how to respect animal welfare. Okay, sure. So all and all cannot be done without the knowledge of genes and genomes. That's the reason we say that the genomics and the foundation of life science biotech by providing knowledge of gene genomes and 3 network. I would like to quote again from James D. Watson. All biology in the future will start with the knowledge of genomes and hopefully proceed. Finally, please allow me to quote from one of our colleagues. Just as physics shocked the world in the 20th century, it is now clear that the life sciences will shake up the world in this century. Now we can hear the steps of this century is approaching. So finally, just for fun, in a sense, just recently, BGI has emerged as a genomic superpower, even though I don't like this term. Now we have so many labs officers in the world. We just opened our lab in Copenhagen, and then we are regarded the bold ambitions of one institute could make China the world leader in genome sequence, even though China is not qualified yet. I don't believe in it, taking the world by storm. We will never take the world. And we are listed as either the 4th or the 5th of the top 10 research institutes in China, even though the others are just working on all the field. We are working on a single field. That's very funny. In 2004, when some people ask, can China overtake the US in science? And the BGI Rise argues Chinese science technology. And now, according to Monitor Group, that BGI may well create a very different model. When I was asked, what is BGI model? I said, no model. Our model, perhaps, if we really try to trace it, rooted in our culture, the first is be persistent. I fully agree with natural biotechnology that the genome sequence would be a starting point to orient and enable all the other hypothesis-driven research. And finally, it will lay the digital foundation for medicine and for life sciences. Second, be humble. China is a little calmer. And then most of Chinese are your students. For example, James D. Watson, and then said that the BGI now compares and competes well with the equivalent center elsewhere in 2000 year. And then China now is a leading genomics nation. And then still many years ago, then I would quote from the Chinese proverbs. Among those 3 passing by, at least there is one who is highly qualified to be my teacher. And there is always another layer of sky above the layer we can see. Be responsible. Be confident. Now we have about 4,000 people. I would show you how tired after they are working instead of how hard they are working. That's in sense the 3rd generation leaders of BGI, and the second leader Wang Jin. And then these BGI researchers are smart, confident for their age, tremendous experience according to a journalist of nature. Then the 4th, a big group of leaders. And then now we are working very hard to educate the young Chinese. I'm confident that's for all best days ahead of us, not behind us. And then this is important. Kind of nothing published by BGI could be done without our collaborators. We just published this paper to call for free sharing. When you drink the sweet water from the well, don't forget who helped you dig it. One day's teacher, all life, that's you. And then a drop of water should be returned with a fountain. I especially appreciate the continuous long-term help from Oz University. Now my former supervisor, Lars Bulund, works at least 6 months, a year for us. Thanks to all the collaborators. Nothing can be done without you. Come to have a look. Come, let's collaborate. Thank you very much for your attention. I have made the mistake again that I have not timed well. I apologize. I thank you again.
Thank you very much, Huanming. I know some of you would have been thinking of questions, but I felt we should allow him to speak. And I think you've heard something very special. It's a new age, and China is contributing to it in such a tremendous way. And I know how much this man has been personally responsible for that. So thank you very much, Huanming.
Yeah, thank you.